dear chairs, thank you very much, dear colleagues. It's a great honor that I can talk to you today about our work regarding the Brainstem Reward Center that we found. And I would like to first thank my students who participated in the project, Krisztián Zihó, Boldizsár Balog, and Réka Sebestény. She's actually also a past NTA Szent Györgyi student, and now she's a Szent Györgyi PhD student in my lab. So we know that the brainstem participates in several different processes, including the modulation of pain, awareness, arousal, and provides the brain with several slow neurotransmitter systems, including serotonin, dopamine, noradrenaline. And it was for a long time thought to be a slow modulatory center for these processes. And my lab is focusing on these brain regions that control emotional functions. But we also discovered in several studies till now, in the last 15 years, that the brainstem can be very fast as well. It produces GABA as an inhibitory and glutamate as an excitatory transmitter, and can reach higher order brain regions, including the hippocampus and cortical regions, with this very fast acting modulation. So this is far from being just a slow modulatory center for brain waves, but it's really a fast control center. You know, if you can think about it, this is also called the reptilian brain of the mammalian brain, the reptilian portion. And reptiles needed to be fast and think fast and had to decide whether something is good or bad for them. This is what actually drives all of us, even today. We have to decide what is good and seek it, and what is bad and avoid it as much as possible. Now, one of these avoidance centers is in the brain is the lateral habenula, which is very important in negative experience processing. This negative experience processing is so important in this region that is actually the major center for depression. So we now know that if the neurons in the lateral habenula are firing bursts of action potentials too much, the animal will be depressed, even in humans. And therefore, we did some studies in lateral habenula, but at this time I was interested in what inhibits the lateral habenula naturally. So instead of trying to target lateral habenula with drugs that are fashionable nowadays, I was wondering what type of innate inhibition it receives. You probably know that there is a method called optogenetics, and in the optogenetics we can use certain animals, transgenic animals, to target a specific type of cell population in the body, in the brain. And also we have specific viruses that can help us doing that. So by injecting, we injected a retrograde tracer virus into the lateral habenula. Retrograde means that it goes back from the axon terminals back to the somata of the neurons. And in these mice, this is the VGAT3 mouse line. This is vesicular GABA transporter, which means that these viruses, because this is a Cre-dependent virus, and that these mice express Cre only in GABAergic neurons, this means that, and because this is a retrograde virus, we could achieve that if we injected this tracer virus into the lateral habenula, it will go into the neurons, but it will express its fluorescent marker only in GABAergic cells. So we could see all the GABAergic neurons that target the lateral habenula. Now, of course, we've seen some other neurons as well in the brain that has already been known. But we discovered that there is a new cell population in the brainstem under the fourth ventricle with numerous cells, and they were completely different from any other known nucleus in the brainstem. We named it subventricular tegmental nucleus because it was under the fourth ventricle. And we also reconstructed it in 3 dimensions, and we found that they are completely different from any other known cell populations, like the dorsal tegmentum, the locus ceruleus, dorsal raphe, and so on. These are the green, this is the green nucleus that we reconstructed from the mouse brain. And if we inject now another virus into the subventricular tegmentum that we discovered that would specifically fill the GABAergic cells only, we could follow their axons, and we could see that indeed their axons reach the lateral habenula, the center of depression and negative feelings. And we found that this is actually the largest purely GABAergic inhibitory input to the lateral habenula. And this might be clinically very important, because what we need to do is to boost the activity, probably, of these cells. So let's see it in behaving animals. I would like to just very briefly summarize several years of studies here, just a couple of important points. One is that we can watch the cells with a fiber optic, and if they are activated, because they have calcium influx, we can see an increase in the light expression, the photon output of these neurons, which this represents, this reddish region represents this light up of these neurons, the activity of these neurons. And this you can see here, when we gave reward to these animals, these cells got activated. If we could activate these cells using optogenetic tools as well. And if we did that only in the striped side of a dual room chamber, then the animals loved this so much that they wanted to stay in that part of the box. Or if we hooked up this system so that the animals could activate their own subventricular tegmentum, because they could poke their nose into a hole which activated an optic fiber that activated their subventricular tegmentum with optogenetic tools, optogenetics and fiber optics. They really loved this. And they addictively tried to poke their nose as much as they could so that they can get their SVTG activated. So this is clearly a reward nucleus, and this is clearly activated during positive feelings as well. We also went further and wanted to take a sample from these neurons and did some RNA sequencing to see what types of transmitters they express and what type of receptors they express, because this would help us understand what kind of other systems are hooked up on these neurons. And without going into details of all the different types of receptors, I would like to just draw your attention to a lesser known transcription factor, which is the SADB1. Now, it is not really important what SADB1 does, but what is important is that in the brainstem, SADB1 was very specific to this nucleus. So we are still in the mouse, but we wanted to know if this new nucleus is also exists in the human brain. And using this SADB1, we could identify the SADB1 positive cells between the dorsal raphe and the locus ceruleus exactly where they were in the mouse, which proves that they are also present in the human brain, which obviously has a very important implication in the future. So as a summary, we found, obviously, we did several other studies, and we found that this new nucleus is driven by the cortex from reward expectation neurons. It is activated and process valence information and activates aversion and memory-related circuits. And it also reaches, it also affects the dopamine release in the nucleus accumbens, which is well known to be a reward transmitter in the brain. Thank you very much for your attention. I would like to just draw your, again, your attention to the fact that this was mostly done by students like you, and obviously would like to thank my collaborators and other members of my lab as well. Thank you very much for your attention.
Thank you very much, Gábor, for the fascinating talk, and also for keeping the time, so we now have some time for questions, if there are any questions in the audience. If not, then I will have one for you. The nucleus accumbens is a very similar center, and it's well known that it is a target of different drugs, which are marijuana, cannabinoids, and so on. And the question would be that if in this nucleus, do you use any size that this kind of psychoactive drugs or addictive drugs also stimulate this nucleus?
Now, this seems to be a very important question because, as you could see in our experiments, the animals really love to self-activate this nucleus. Although I am not sure how specific it is for this nucleus. As the brain is wired, it really does two things. It tries to avoid things that it doesn't like and wants to get things that it likes. And anything that is pleasurable, the animals or even humans would like to seek and get as much as they can if it is not harmful for them. This was the reason why we decided to check the messenger RNA content of these neurons, so that we can map all the different receptors that are expressed in these neurons. And we've seen opioid receptors, several of them in these neurons. So I guess that this might be a site of action for opioids as well. Although it's very hard to judge, right? Because sometimes it also depends on the balance of the whole nervous system, how it acts. Like this is the same story as with the SSRIs and the serotonin that was for a long time thought to be one of the most efficient drug against drug against depression. But obviously, the whole brain is full of serotonin receptors. So this is kind of a sheer of luck that the activity of these receptors in certain parts of the brain that are more pleasurable or more balancing is a little bit higher than in other parts of the brain that has something to do with memories, say. So that's the reason why we are doing this kind of research. We would like to map different parts of the brain, different nuclei in the brain. So in the future, we do not just have a personalized medicine, but a brain region or even a cell type specific medication and targeting, either with the gene therapy or with other methods that can specifically target certain cell populations in the brain. And this is also a hope for, in this case as well.
Okay, thank you very much. Are there any questions? If no, I would like to ask you to join us for a group photo.